Okay, I am now recording. If you can hear me, write 1 in the chat. Okay, good. There's some other people, I presume they're just going to catch the replay online. So here we go. So the gist of this is, this was a request lecture or a request lab that I could quickly go over, maybe like the top 10 things that I'm doing most commonly in bioinformatics. These are all really, really easy things that you guys can do. They don't require any computer coding. They just require a moderate understanding of the central dogma of biology. Anybody can do this. And so this might be too basic for some people, but it's probably worth catching up everybody to the same level so that we all know sort of like what's out there, what you can quickly do, and what I find myself doing very, very commonly and very often. Okay, so the first website you definitely want to know about, if you haven't heard about it yet, is the National Center for Biotechnology Information. So if you go to this website, ncbi.nlm.nih.gov, this is NIH funded repository of information. Every single genome that's ever been sequenced by NIH funded money is posted to this website. I'm trying to optimize the camera. Sorry, I was debating whether I want my picture in the recording or not. Well, I'll just leave it in there. Okay, so this database, you can do a lot of things in this database. You could certainly spend a whole class describing all the things that you can do on this database. Okay, so what I am most commonly coming here for is number 2, finding a gene X's sequence. Okay, so it's a very, very common thing in biotechnology where you're going to study X. The first thing you need to do is grab the sequence of that gene X. Okay, you can do that at NCBI provided that the genome has been sequenced. So we just gave a lecture, which I'll post, on P elements. So for an example, I'm going to see if I can find the P element sequence on this database now. So what you would do is you go to the top. I'm going to type in the name of the gene. I know what it is. So I'm going to say P element transposase, and I know what it's from. It's from Drosophila melanogaster. There's going to be many different orthologs of this P element transposase. I want the specific one from Drosophila melanogaster. I want to see its sequence. Okay. Now you can select different databases. I know what I'm looking for. I'm not looking for a genome. I'm not looking for a protein. I'm looking for the gene sequence. I want the gene. So I'm going to try searching gene and just see what happens. There it is. Interesting. What we're seeing here, wimpy testes. This is funny that I did this. In the lecture, I talked about how you get hybrid dysgenesis from the P elements. There's probably some phenotype where they're sterile or something, and they've named it wimpy testes. But peewee, this is not the gene I'm looking for. Interesting. So these are the names of the things that came up when I searched this. Peewee is not the gene I'm looking for. So how could I adjust this? Let's just try transposase, Drosophila melanogaster. I hope this doesn't end with an abysmal failure. Maybe it is this one. Let's take a look at it. So if I click on it, the peewee, let's just see what it is. I'm not sure if this is the actual transposase or not. There should be 4 exons. Nope, definitely not it. So I clicked on the peewee. It's worth going over what it's showing us here. Again, I look at this all the time. So at the top of the page, there's like stuff to look through, information. This is the organism. This is the chromosome. So it's on chromosome 2, and the L is a particular site of the chromosome. Okay. And here's the actual gene. These are the genes that flank it. This ABO, ATP, SYNG, SCAR, CG, 1, 2, whatever. These are genes that flank it. Okay. Now DNA is directional. So this gene on this chromosome is apparently going this way to the left. So that would mean that the coding strand of the sequence is on the other side of the DNA. So if I look down here, this is the actual gene. And now you'll notice that it's pointing to the right. Here it's pointing to the left. Here it's pointing to the right. So what it's showing me down here is the 5 to 3' sequence. This, it's showing me the 3 to 5 orientation. Okay. So the green things, this is a eukaryotic gene. I can tell it's a eukaryotic gene because I can quickly see these dark green boxes. These are the exons. So this gene has 1, 2, 3, 4, 5, 6, 7, 8 exons. Maybe these lighter ones, I'm not sure what these are. These might be alternatively spliced exons, or it could be the terminator. Looks like this is a promoter region, this light green thing, or it could be that this is the start codon. If we want to zoom in and look on this, let's zoom into this region. So sometimes what you can, you can just like highlight. Let's see if I can do this. Zoom on that range. So this is those first 2 exons. Let's see if we can find the start codon. If we keep zooming in, I'm just clicking on it now. Let's hit the plus to zoom in. We should eventually be able to see the DNA sequence. Yeah. Here, so this is the start codon, ATG, right here. So this dark green with the white arrows is the exon, and it looks like this one was the promoter, as I thought. So you can actually zoom into the point where you're seeing the sequence. Let's zoom back out. Okay, you just click the minus thing to zoom out. And you can zoom out so far that you can start to look at things that are around it. So again, these are those flanking genes. You can move to the right or to the left. And this is a very, very complicated viewer. What you're seeing are down here are different proteins that are probably being synthesized, alternative proteins that are derived from this ORF. This is probably the expression data. Yeah, RNA-seq. So Drosophila has very, very advanced data with every gene. These are expression data that show you when this thing is being expressed, how much it's being expressed, what the codons look like, things like that. Okay, so I'm very, very commonly looking at genes in this viewer. Okay, I didn't actually find the gene I wanted to find though. So let's go back. Let me see if let's try once more to see if I can find P element, and then we'll move on if we can't. I mean, I should be able to do this. P element transposase. Let's just try Drosophila. Maybe I'm being too specific. Repressor of the inhibitor of the protein kinase like. I know it has a THAP domain. Let's try THAP. I'm going to try this thing. This is from a different species, Drosophila heidii. I'm going to see what happens when I click on this. I'm going to see what it looks like. I know the P element has 4 exons. That's what I'm looking for. No, not it. Okay, let's see if we can find a different gene. Oh, something in the chat. What does it mean if a sequence is labeled as variant? That means there's either splice variant, there's probably splice variants that generate different proteins. It's very common in Drosophila. You might have one ORF that can generate 10 different protein variants with different splices. Let's try a different gene. Let's try ricin. Ricin is a toxin from bacteria that can kill people. I'm teaching people how to search for ricin. Here it is, from the castor beans. Okay, so let's look at this peptide toxin. So here's the gene for ricin from castor beans. I'm zoom out. Oh, okay, so this is interesting. So this genome of this organism must not be sequenced because it's only showing me a partial sequence. It's not aligning it on a whole chromosome. Yeah, and here's where it says unplaced scaffold. That means this genome has not been sequenced and assembled. Let's try something different. Here, this one looks like it's actually been assembled. we're seeing many different orthologs for ricin. Okay. I don't know if I should, I'm going to get called by Homeland Security. I have to pick a different gene, because if I teach you guys how to clone this, I'll get called by Homeland Security. What would be a different gene? Let's try actin from Drosophila. Actin Drosophila. Here we go. Okay, so there's many different actins from Drosophila. That's kind of like the hardest issue that you need to solve when you're searching for gene X. The biggest issue is many of these genes have many different names. Look at all these different names associated with this one gene, okay? And the hardest thing is matching the right name. And so when I was searching for P element, that's the colloquial name. Maybe perhaps when it was filed in the database, it wasn't filed with that name, so I can't find it. And so you have to get the name right. That's the thing. And that's the importance of looking in the literature, figuring out what people are calling it in the literature. But here we're able to find some different actin paralogs. So if they're all from Drosophila melanogaster, so this is an ACT5C paralog, ACT88F paralog, actin, ACT42A. Let's click on one of these, and I'll show you now how to pull the sequence. Okay. So now if we find a gene, if we actually find a gene that we're looking for and we want that sequence, the way I get it is I go to the gene and you can right click on it and you should be able to, maybe not right click. If I hover over it, this box pops up and then I can essentially FASTA record. If I click on that, it should literally give me the sequence of this gene. Yep. So it's now giving me, this is the sequence, and it's giving me actually this base pair to this base pair on that chromosome. Okay. And it doesn't start with ATG because it's giving me some of the promoter. Okay. So if I wanted to pull this sequence and clone this gene, I could directly pull this sequence and paste it into my Word document, and then start looking at it there. I wouldn't usually look at actual sequence here. Let me actually show you now the difference between like a bacterial gene. So the eukaryotic genes are going to be a lot messier because they're going to have long promoters, they're going to have long terminators, they're going to have introns and exons. It's going to be really, really messy. If you study bacteria, it's going to be real nice. So let's look for like Xanthomonas Virdi4, which is going to be in the type 4 secretion system. Wait, I spelled it wrong. Xanthomans. Let me fix this. Xanthomonas. Let's say I want Xanthomonas Virdi4. It's a name from a bacteria gene. Type 4 secretion system conjugate DNA transfer family protein. So it wasn't actually, see, here's what I'm saying. It wasn't actually named VIRD4. In this organism, it was named HLH45RS21510. So sometimes it's hard to find the thing you're looking for based on name alone. And so that's going to bring us to our next thing that we look at. So let me see if I can grab this gene and I'll pull it. You'll see how bacterial genes are different. So bacterial genes should have no exons or no introns, should just be one big exon. And that's exactly what you're seeing with this green bar. Okay. So most of the time I work on bacterial genes, which I love because they're so simple. It's really easy to work with them. So let's try to grab this sequence. Let's hit the FASTA record. That should pull up the gene for the VIRD4 from Xanthomonas. And here you'll see, now you're seeing the start, the ATG. So again, bacterial genes are very, very simple. So you're always will start your FASTA sequence with the ATG, the start codon. At the end here is the TGA. So if I wanted to clone this gene, I could design my primers from this sequence directly. And what I would do is I would copy it and I would paste it into my Word document. Now, one of the things that happens when you paste this into Word documents is Word will automatically add a new line entry. And so I have to go through and manually delete all those. I'll show you a faster way to do this at the very, very end. but I will usually run through my sequence and get rid of all those new lines once I paste it into the Word document. Because if I ever want to search for something, a pattern in the sequence, that new line, that carriage return will cause a problem in the searching algorithm. So I always remove these. And then now imagine you wanted to search for, oh, where's the start codon? I can control find ATG. And it shows me, okay, these are all my potential options for the start codons. It's obviously this one because it's right at the beginning. Okay. So if I wanted, I might as well go into this into great detail, I guess. This would be, I would use this if I was cloning and designing primers directly from the start codon and the stop codon. I might not want to do that. I might want to give myself some extra space. So if I want to go into the promoter or the terminator, I can just change these numbers, the selected region, okay? I can change this number, I can reduce it by 100, and it's gonna go 100 base pairs extra to the 5' side. And I can increase this one by 100, which is gonna cause it to become 6029. And so now if I update the view, now what it's giving me is it's giving 100 base pairs of the promoter and 100 base pairs of the terminator. So perhaps you want this instead, and then I would mark where that start codon is. Okay, so you can adjust what you're grabbing, but this is essentially how you're pulling sequences from the database and how you're designing primers for PCR, which we're going to go over in the next coming steps. Okay, so that's the first two things. Let me introduce for you the conserved domain database. Once we have the sequence, we can do a lot of different things. One of the most common things I'm looking for as a molecular biologist is the first thing when I'm looking at any protein, the very, very first thing I want to know is what's in it? What kind of a protein is it? So if I go to this was that Xanthomonas type 4 secretion system gene, okay? I'm going to go back to that original FASTA record. And I want to try to find the protein accession for this. I might not be able to get it because we were searching a protein. Okay, so let me jump ahead and show you what let me see what happens when I click protein. I think this is the protein. I think I just clicked that protein button and it gave me the protein that associates with that gene. Let's test that. And the way that we can test that is with the conserved domain database. So when I have a protein accessed, and you can tell it's a protein because here the sequence now, the tab says protein. If you click identify, whoops, something clicked funny. If you click identify conserved domains, this is going to access the conserved domain database. And this is essentially going to tell me like, what is the protein? What identifiable domains are there in this? This protein has no conserved domains. Okay, so bad example, or perhaps a good example, because this commonly happens with bacteria. Let's pick, let me search for a protein called vir D4 Xanthomonas. Now I'm searching protein, not gene. Okay, so here's vir D4 from Xanthomonas axonopodis, some strain. So it's 565 amino acids. Let me now go to the conserved domain database, identify conserved domains. Here, now it's seeing a conserved domain. So the protein is one amino acid here, the N terminus. Here's the C terminus, 565 amino acids long. This is the vir D4, the conserved region. So this is a homolog of vir D4, and the algorithm knows that, and so it's labeled this conserved domain vir D4, and it says vir D4 is a part of the type 4 secretion pathway. So I know by looking at this that this protein is important for secretion. Let's look at something like the actin protein, or let's look at a transposase. Let's just search this. IS1 family transposase. Let's look at like what a transposase looks like. Okay, so this is a protein. It's a transposase from a plasmid of Salmonella. Let's click on this. And then let's click identify conserved domains. And this should tell me like what domains are in a transposase. So here's a DDE, that's probably catalytic residues. And it says IS1 superfamily transposase. Transposase proteins are necessary for efficient DNA transposition. This family represents a bacterial IS1 transposase. Okay, so essentially what you're seeing is the algorithm is identifying conserved domains. And this is very, very useful to identify conserved domains. And I'll do this a lot because, like I said, if you click on a protein, I want to know what's in it. And these two examples have been kind of poor examples because they've been very simple proteins where you just see kind of like one conserved domain. Many proteins are chimerics of many different domains. And so this might have, in a big protein, this might have like 20 different domains. And that will really give you an idea of what that protein is kind of like doing. So I do this a lot and I look at these data a lot. Now, the next thing, Interpro Scan, this is very useful for identifying conserved domains in proteins that haven't been annotated properly. So a lot of times when you study bacteria, as you just noticed when I clicked on that one random protein, you'll click on the conserved domain database and it'll essentially say there are no conserved domains identified. That means this protein is not similar to anything else that's been studied. So what do you do when you're at that point? What do you do when you have like nothing to go off? That's when I usually use Interpro Scan. So Interpro Scan is a way where I can take a sequence, okay, and I can enter that sequence and it can sort of use better algorithms to try to identify distant domains that might be homologous. So let me try to get an example of this. Let me see if I can pull up a document that I maintain of the proteins that I study. So I'm just going to use this as an example. Okay, so this is one protein that I study. Its name WPIP0283. This is a protein that we're going to cover in detail when I talk about CI. This is its amino acid sequence. And when we first started studying this, nobody had studied it before. Okay, so we couldn't really figure out like what it does just based on looking at the database. So then we would take this protein, we would go to Interpro Scan. So if you just go to Google and you search Interpro Scan, you'll come up to this website and it'll ask you to just input the sequence. Okay, so I can literally just paste the protein sequence, the string of the protein. This is the string of the protein, and I can just click search. And what this is going to do is it's going to try to identify domains, protein domains that are a part of my protein. Sometimes it might take a little bit. So while it's searching, we'll come back to that. Let's talk about BLAST-P and BLAST-N. So what was this gene? This was the type 4. Okay, this is the type 4 secretion system. Let me get that. Let me see if I can get this protein sequence. I'm just trying to grab a sequence here. Actually, I'll just use my protein. You can use any protein to experiment with this. I'll use mine because I know what the results should be. So I'm going to copy my protein string. Now I'm going to teach you guys about BLAST. Okay, so if you go back to the NCBI homepage, a BLAST is essentially, I'll draw this out here. A BLAST is essentially like if you have an ortholog and you want to know, does organism X have an ortholog of gene Y, you can use BLAST to test that hypothesis. So if you're studying the Xanthomonas gene, you might want to take that Xanthomonas gene and see if E. coli has the same gene. And then you could look at all the papers in E. coli that have been studied on that particular gene, and they might be useful for your Xanthomonas research. So this is a very, very common thing you want to do, and you want to use this in BLAST. So you go to the NCBI homepage, and you click on BLAST. BLAST is going to allow you to do that search. Now you're going to have various options, okay? Nucleotide BLAST, these other options, and protein BLAST. I'm doing this simple and fast So most of the time, you're either going to be just doing nucleotide blast or protein blast. So let's do protein blast because I told you in the previous lecture, most of the time you want to blast proteins because proteins are the most conserved. There's a lot of wiggle in the nucleotide bases at the wobble position that allows mutations to occur. So most of the time, if you want to search for a distant ortholog, you're searching for blast P. Okay, so we have blast P. Now I'm just literally going to paste in my sequence. This is my protein, my special protein that I study. Okay, and now we can do two options. If we don't type anything into the organism, it will blast this protein against everything in the entire database, and it will give me the closest hits. Okay, so if I was to blast this and not type anything in the organism, let's see what happens. I'll click blast. Now this usually takes a little bit because it's running a bioinformatics algorithm. So while it's running, let's transition back to the Interpro scan. Hopefully that one is done. It looks like it got results, and it looks like I have to click on them. So it's got results from the Interpro scan of my protein. Let's click on that. And here it's identified some domains. So here's the N terminus, here's the C terminus. And it looks like it's identified, yep, this makes sense. So it's found I don't know what it's being funky, but it's identified peptidase C48. That makes sense. My protein that I study is a peptidase. It's a deubiquitolase, so it cuts proteins. So it's found this family, and I can probably click on this, and it highlights exactly in the sequence where it's finding this domain. It's saying this region right here is a C48 peptidase. Where'd it go? Oh, shoot. I clicked a button. Here we go. And it's got these other domains. So these are other just ULP protease. These are just other protease domains. Papain, like that's another protease. Adenoviral proteinase, that makes sense. It's a protease. So it's saying this region of the protein is similar to all these things. Okay. And it's saying this region is a disorder prediction. So if you ever get in a scenario where you have a protein that you want to study, but it has no conserved domains, is not similar to anything else out there in the database, and you can't find any conserved domains in the conserved domain database, run an Interpro scan and see what Interpro scan pulls up. And you can, all these things are clickable, like you can click on them and they'll tell you like about the thing you've just pulled up. Okay. So I do this with proteins all the time. Let's go back and see if our blast is done now. Okay. So here's our blast. Now I didn't type anything into the organisms. So it blasted against all the organisms in the database. And it's saying the closest match is a deubiquitolase from Wolbachia pipientis, which is true because that's what it is. It's exactly what it is. I identified it. That's what we characterized it as. Okay, so it found it. And these are all the different orthologs that it's similar to. They're all from Wolbachias. That's the organism that I study. That's where it comes from. So all the things that it's similar to are from Wolbachias from various insects. Here's Wolbachia from a Drosophila. Here's an ortholog from Wolbachia from whatever this thing is. Here's an ortholog from Wolbachia from Chrysomya. That's probably a honeybee or a bee or a bumblebee. So all these are different orthologs. Now, if you click graphic summary, it should give me that color scheme. So it found a whole bunch of red hits. These are like 99.9% the same. And for each of these hits, you can click on it. Let's say you want to see this hit. And it should literally pull up maybe it's not doing that anymore. You got to click the alignments tab. If you click on the alignments tab, it'll literally show you the alignment. So it matches this protein with 100% identity. That means it literally is this protein, this deubiquitolase from Wolbachia. Let's check out some of these other ones. This one, it matches with 99%. And if you want to find where the mutations are, you look for these regions here. So it matches this one, it's conserved, but there's a little bit of a mutation spot here where it's got a lot of differences. And you can see that because there's these empty spaces in between. And as you move down further and further and further and further, you're going to be looking at more and more and more distant homologs. So you're going to see these distances increasing. So you're going to see more spaces, more of these plus signs. A plus sign means it's an amino acid that's similar, but not the same. Okay. And an empty space means like this is something completely different. This is amino acid that's completely different. This is a deletion. So if you see a hyphen, that means this base is deleted. These bases are deleted. And again, if you move more further and further and further and further down, they're going to get more and more and more distant based on that graphical summary. So this is what you would do if you want to look for what's the closest thing out there? What's the closest thing in the database? Let's go back. Let's edit the search. Now imagine a scenario where you want to know, does organism X have gene Y? So I want to know, does E. coli have an ortholog for my protein? If I want to know that, what I can do is I can search, I can type in the organism, Escherichia coli. And it has a bunch of taxids, so there's a bunch of different E. colis. Let's blast it against all E. colis. That's what essentially this is telling it. This is essentially saying, check this protein and see if E. coli has this protein. So let's click blast. And let's do that. Okay, while we're waiting, let me look at this. What's next? Okay, so we covered blast P. Now, blast N is the same thing, except you're just searching nucleotide sequence. So if you want to see if it has a nucleotide sequence, you would enter a nucleotide sequence. So that's the same kind of thing. It's not much different. I'm debating, do I wait for the blast to finish or to run into Expasy Translate? While we're waiting for the blast to finish, let's discuss Expasy Translate. Let's discuss Expasy Translate. So oftentimes, if I grab a sequence, I don't even remember what the sequence was. I don't even remember what it was, but I grabbed it. Oftentimes when I grab sequences like this, I want to get the protein. So you can just use a computer to translate this. The computer will act as your ribosome. So if you go to a web browser and you just Google Expasy, E-X-P-A-S-Y translate, you'll come up with this server. You can literally just paste your DNA sequence, okay? And you paste your DNA sequence in, and you can click these different buttons. Let's click Compact M, and then you just literally hit Translate, and it will take these codons and translate it, okay? So there's going to be 6 different frames, 3 reading it from left to right, and 3 reading it from right to left, okay? And the first frame is going to start literally at the first base. So it's reading it starting here. The second frame is reading it starting here. And the third frame is reading it starting here. The 3 different reading frames, okay? Now, I know which one is the obvious reading frame because it's pink. There's no stop codons in it. So Expasy Translate is highlighting open reading frames in this pink color. So if you see a giant open reading frame like this with a methionine at the beginning, the hyphen is a stop codon and a hyphen at the end, you know that this is the protein. So I know that this is the protein. So whatever this thing was, whatever this was, this is its protein. And I know that this is not its protein because it's a tiny sequence with a stop codon, and there's a bunch of these other tiny sequences with stop codons. So if you just translate random DNA base pairs, you find stop codons all over. That's how you know it's a gene. It's a gene if you take the sequence and you can translate it into a frame that has no internal stop codons until the very end. That's how you know it's a gene. And that's how you know that this is the sequence. Now, you might want to optimize a few variables for this. So oftentimes a question I want to answer is, if I want to make a mutation, sometimes I want to answer, where is codon X. So imagine I have this sequence of the protein, and let's say I want to target this methionine for a mutation. I need to be able to find this methionine in the DNA sequence. So I need to know, where is codon X? Where is the codon for this methionine? You can do that with Expasy Translate, okay? So if you click the button includes nucleotide sequence and hit translate, now it translates it, but it also includes the nucleotide sequence. So now I can use Control Find to find the codon. So if I wanted to find this codon, I know that the sequence goes DGGM. So let's Control Find DGGM. There it is. This is the actual codon, the precise one that I wanted to find. This is its codon, ATG. Now, if I want to go back and find that in my sequence, I can just take this, okay? Now I can Control Find it in Word. I have to delete the spaces. But once I delete the spaces, it should find it. There it is. Here it is. So we just found a very, very specific codon which codes for that precise methionine. So I know if I wanted to mutate this methionine, the bases that I would target is this ATG right here. So I would design primers based around this. We'll cover that in site-directed mutagenesis in the future lectures. This is how you would find the codon. So that's another common thing I'm doing with Expasy Translate is, where is codon X? So Expasy Translate is used for, where is codon X and what protein does this sequence make? Okay, let's check our BLAST. Okay, so our BLAST finished and the result was no significant similarity found. Okay, so what it did is it searched the entire E. coli database for an ortholog of my protein, and it didn't find one. That means E. coli does not have my gene. That's useful information to know. Okay, so if it's not there, it won't find it, and it'll tell you that you didn't find it. Okay, so that's the gist with BLAST. That's the gist with Expasy Translate. These are done. Let's quick go over Clustal Omega. So Clustal Omega is just like a common alignment program. So the situation under which you would use this is if you have a list of orthologs and you want to build a tree, or you want to align at least 2 to maybe like 10 orthologs. If you want to like align a bunch of orthologs, this is what, I don't have an alignment pulled up. An alignment was that thing where it was comparing the residues. If you want to do that, go to Google, search Clustal Omega, which is a multiple sequence alignment program. You have to set it to either protein or DNA or RNA. So typically I'm aligning proteins. Let's do a test. So let's align my favorite protein, WPIP0283. Now you have to put a caret and then paste the protein. This is what's called FASTA format. You put the caret and then you paste it. And then we need a second thing. We need at least 2 things to compare each other to. So let's put a second caret and let's paste the same sequence, only this time let's make some mutations. Let's make mutations. Let's make at this LVD, let's make a mutation and let's make it alanine, alanine, alanine. So this is a situation where you might have 2 orthologs or more, and you want to see how are they different? How are these orthologs different? And then you literally just hit submit. Uh-oh, error in reading the sequence. I did something wrong. Let's try removing the carets. And we're just separating these sequences by a carriage return. Let's try this. That worked. Oh, no, a minimum of 2 sequences is required. Okay, so something is wrong. Let's try caret return. caret return. Let's try this. Nice. Okay, so it was, the key was caret return. So the FASTA format is technically caret, and then there's usually like name X, and then it's preceded by sequence onto N. That's usually typically FASTA format. So the key there was doing the caret and then the return. Here's what it spits out. Here's our alignment. If you see a star, it means it's the exact same amino acid. So here was the first thing I put in. Here was the second thing I put in, and it quickly found the differences. The differences was where I changed the LVD to an alanine, alanine, alanine. So this is very, very useful for aligning multiple proteins. If you ever want to answer the question, of like, how are two orthologs different, or what does the evolutionary tree look like for a set of orthologs? That's what you're going to be using Clustal Omega for. And you can quickly click on the tree, show phylogenetic tree. Now, I only put in 2 things, so this tree is kind of pointless. But if you had like 12 different things, it would literally plot you a cladogram of a phylogenetic tree of a bunch of different orthologs. So you could actually look at the evolution and try to make inferences to the common ancestor based on these phylogenetic trees of a set of orthologs. Okay, so that's Clustal Omega. Almost done here. We weren't waiting on anything, right? No, we weren't. Okay, so the final 2 things I'll show you that are very, very useful for bioinformatics are Word. So this might seem counterintuitive. I love to play with sequences, DNA sequences in Microsoft Word. I am always pasting DNA sequences and control finding. So imagine I wanted to find this particular sequence. If I have that sequence in Word, I can control find, paste the sequence, and it will find it for me. Very, very commonly, I'm making documents like this where I'm collecting information about proteins. I'm marking very specific amino acids because they might be important. I'm marking patterns. So I use Word all the time to sort of like make my personal annotations and make personal notes to myself about the genes that I'm studying, and I keep track of them. Word is like a great program. And there's a lot of people who don't use Word, but I would really encourage you to do it because one of the reasons is it will force you to understand 5' to 3' directionality. It will, because it's only showing one strand of the DNA, it will force you to figure out like what direction are you looking at. Whereas if you're looking in like a DNA viewer, it might automate that. So you might not know what direction you're looking at. Word kind of forces you to understand that. I design all my primers for PCR or mutagenesis or cloning. I design all my primers in Word. And the reason that that's good is because it forces you to figure out, it forces you to look at the sequence, find, okay, what are my CG clamps going to be? Maybe it's going to be this. It forces you to pick out a sequence and then figure out how to analyze the sequence in a way to determine if it's going to be a good primer or not. A lot of people don't use Word. A lot of people will use like automatic PCR primer designs. That's a terrible idea because then you don't learn how to actually like look at the sequence and figure out like what would be a good spot. You're relying on a computer algorithm to think for you. That's always a bad idea if you don't understand the basics. So I love Word for DNA analysis, and I would really encourage you as you move up in your careers to really push yourself to use text editors where it forces you to understand what you're doing. Okay, so that's my gist with Word. Now, the final thing I use is a special text editor called Sublime. So this is a free, don't look at this, this is an email list. This is a free text editor, which is very common for computer programmers. I use it a lot. And the reason I use it is because it has really, really, really easy, what are called regular expressions. So let me define this. You'll see this abbreviated. It's coding language. It's actual like computer coding language. And it stands for REGEX, stands for regular expressions. And regular expressions is kind of like a code that you can type in that allows you to identify specific sequence elements. And this is very, very, very useful for large data sets. So let me show you an example of this. Imagine you had a data set that looked like this. There was a tab and there was like name X space, and then let's say sequence. And then there was ortholog 2 name X sequence. And then there was name X sequence. Let's copy paste this a couple times. Oh, hang on a sec. In bioinformatics, there are many situations where you will pull data from a website or a server, and it will be in a very, very strange format. There will be lots of underscores. There will be lots of money signs and dollar signs and stars. And there will be things that make things different from each other. And there will be some common elements like tabs, and there will be some common elements like spaces. And there might be a situation where if you have, say you have 10,000, imagine you pull a database and there are essentially like 10,000 of these things. And you need to optimize the data for a particular input, okay? Now, normally that would require you to use Python or actual like coding language to do that. But if you have Sublime, you can use regular expressions to quickly like pull out certain or delete certain spaces. So when I'm in Sublime, let's say I want to get rid of the tab. Let's say this tab is extremely problematic for me. It's causing a formatting issue. Like you just noticed that there was a formatting issue when I was trying to create an alignment where I needed all my alignment things to look like this, where there was a caret, a return, and then the sequence. That's a very, very common situation in bioinformatics where a lot of it is like dealing with formatting issues, computer formatting issues. So if there's an issue like that and say you need to get rid of 10,000 tabs, one way you could do it is you could go through the entire list and detab every single 10,000. Obviously, that's a terrible way to do it. So if you have Sublime or a text editor, you can hit Control H. That's going to pull up a find bar, and you can find tabs. There's a code that means tab. I think it's back. What is tab? I don't even know it. Is it? Let's search it. So if I don't know the code for a particular character, I search what is the regex code for tab. And it's I had the wrong backslash. It's /T. So it's /T. Now, when I tell it to find all those tabs, it's found every single tab in the entire sequence. Okay. And if I wanted to delete that, I put nothing in replace and I just hit replace all. Boom. It deleted 10,000 tabs. Okay. So there's also situations where you might want to extract a particular element. So imagine I wanted to extract 10,000 sequences from this entire thing, okay? One thing I could do real quick is I can type tab name X space, okay? And now I can replace that with nothing, replace all with nothing, okay? And now I'm going to use regex codes to try to find the rest of the lines. Now I have extracted 10,000 sequences and gotten rid of all the extra crap. Okay. So this is essentially like an example. You are not going to learn how to code unless you take classes on it. And regular expressions is sort of like a little trick that can kind of get you halfway to coding. And it can get you like half of perhaps what a bioinformatician could do with maybe like a Bash program or a Python program. But you can do a lot of stuff with it. And most of it I just learned by just Googling like regex codes for Sublime. And you can essentially like, so let's see if we can find, yeah. So here's all the codes that allow you to find certain elements, and you can make different combinations of these things to pull out or extract particular things. So again, I'm not going to, the point of this is not for me to teach you regular expressions. The point of this is for me to teach you that they are available. And if you ever get into a situation where you have a database that looks like this, and you need to extract a particular element or delete a particular element, you can use regular expressions to quickly figure out how to do that, and to do that to save yourself from having to delete a million things individually. Okay, so that's kind of like the lecture, a lab lecture on just kind of like basically top 10 bioinformatics things that I find myself doing all the time. If you master these things, you'll have a really great subset of bioinformatics skills, which then you can build on. Now, I'm not talking about any of the specific, like more specific algorithms or programs or tweaks to BLAST that you can do, all the things like that. I'm just giving you the basic skill set with which you can build. Okay, so are there any questions? If there's no questions, you can type them in the chat. Otherwise, we can conclude the lab there. Okay, so I will conclude the lab, and I will see you guys on a Wednesday. And I will post the lecture and I will post the lab so that people who missed it can catch it. Okay? Have a good night. I hope this was helpful. I hope this satisfied the request of the top 10 bioinformatics. Let me know if it did not. If there's anything else you want to learn, just let me know. Okay? Bye-bye.